Hello everyone, I hope you are doing well. This is section 2 on development of tooth. If you haven't watched the section 1, I request you to watch the section 1 first and then continue with section 2. Please watch the complete section. In section 2, we are going to do a rapid review of section 1. In section 1, we discuss the formation of dental lamina and vestibular lamina. In vestibular lamina, there is programmed degeneration which results in the formation of vestibular cleft. Similarly, there are changes in connective tissue such as condensation of connective tissue cells. These connective tissue cells are derived from neural crest cells. These are the very important topic to remember. The connective tissue cells are derived from neural crest cells and the programmed degeneration which results in the formation of vestibular cleft. These are the important topic to get remember. It might come as MCQ or may ask as Viva. If you have any doubt, please go back to the video and check. If you are interested in watching the complete section, the link is available in the description. Please go and check out. In this section, we will study the bud stage and cap stage of tooth development. We also discuss the transitory structure that are formed during these stages. Bud stage of tooth development. In bud stage, the epithelium further invades into ectomesenchyme, giving a bud-shaped appearance. I had told in the previous video, in the first section, the dental lamina. Here what happened in bud stage, this epithelium will invade into the ectomesenchyme, giving a bud-shaped appearance. You can compare this picture with the picture of a bud of rose. It resembles similar like a bud shape. Here you can see the dental lamina. Inferior part of the dental lamina forms the bud-shaped structure. In bud stage, there is little changes in shape and function as compared to the dental papilla. Therefore, these cells are poorly differentiated. In mesenchyme, here you can see, here the mesenchyme, the cells are densely populated around the epithelial bud. This epithelium and this connective tissue is separated by a basement membrane. This basement membrane separates the epithelial bud from the surrounding connective tissue. Next is cap stage of tooth development. In cap stage, there is differential cell division in bud. This means there is unequal cell division in different part of the bud. There is unequal cell division at the different part of the bud which will leads to the formation of a cap-like structure. You can see the picture here. It exactly resembles a cap. At this stage, you can see a variety of cell type. You can see here there are variety of cell type. The cell that is present at the center of the cap are different from the cell that is present at below the cap. And there are also different in cells that is lining the cap. And there are also cell outer surface of the cap. This phenomenon is known as histodifferentiation, means there is differentiation in cell. You can see here the cell that is present in the center and the cell that is present just below the cap is different. Even at the outer surface, the cells are different. In cap stage, the cell that is present just below the cap is known as dental papilla. These are the dental papilla cells. There are also ectomesenchymal cells surrounding both the enamel organ and dental papilla. That is known as dental follicle cells. So this is the dental follicle cell. This is the dental papilla cells. This dental papilla cells will later differentiate and contribute to the formation of dentine and pulp and the dental follicle cell that helps in the formation of supporting tissue of the tooth, including alveolar bone, periodontal ligament, and cementum. Now there is a term called tooth germs. Tooth gum consists of enamel organ, dental papilla, and dental follicle. Together, this structure is called as tooth gum. In cap stage, the cells that is present in the center of the enamel organ. Initially, these were not stellate reticulum cells. Initially, they were polygonal cells, and they were attached to each other by desmosomes. These cells begin secreting glycosaminoglycans, which is hydrophilic in nature, means that attracts water. It's a water loving. Because this glycosaminoglycan attract water, water starts accumulating between the cells in intracellular spaces. As water accumulates, the cells are pushed apart and become star-shaped. The cells that remains connected only through desmosomal attachments. Now, this star-shaped structure is known as stellate reticulum cell. Now, there are two other important types of cell that is present. The cell that is lining the outer surface of your enamel organ towards the dental follicle, that is known as outer enamel epithelium. The cell lining the inner surface of your enamel organ towards the dental papilla cell, this is known as inner enamel epithelium. And there is a basement membrane that separates your outer enamel epithelium from the dental follicle cell, and there is another basement membrane. You can see here in the picture there is another basement membrane that separates your inner enamel epithelium from the dental papilla. So now in this picture only you can see varieties of cell. So there is varieties of cell means histodifferentiation is there. So what all cells are present here? The first one is stellate reticulum. The second one is dental papilla cell. Third is dental follicle cells. And here outer enamel epithelial cells and inner enamel epithelial cells. Transitory structure during the cap stage of tooth development. The first one is enamel niche. In certain sections, the dental lamina may appear as double attached. The dental lamina is not always a single rod-like structure. Here in the picture, it is shown as single rod-like structure. It might be there will be 2 or 3 attachments. Between this attachment, there is connective tissue present. The central connective tissue is filled with the filled area that is known as enamel niche. Next is enamel node. Enamel node is a cluster of cells that forms a bulge within the enamel organ located near the inner enamel epithelium. This is the enamel organ that is present very close proximity to your inner enamel epithelium. This cell may function as signaling center by expressing the growth factor that helps to transform the cap stage into your next stage, that is bell stage. You can see enamel knot is present very close to your inner enamel epithelium. And these enamel knots are usually present at your incisal edge or cuspids. The last one is enamel cord. Enamel cord consists of strands of cells extending from your enamel node to outer enamel epithelium through stellate reticulum. This is your enamel cord that extends from your enamel node to outer enamel epithelium with the help of the stellate reticulum cell. This structure is also known as enamel septum because it forms a bridge between your enamel node to outer enamel epithelium. It may provide a continuous supply of cell to enamel node to outer enamel epithelium. So these are the 3 transitory structures. The first one is enamel niche, second is enamel node, third is enamel cord. This marks the end of section 2. In next section, we will study the best stage of fruit development. Thank you very much for watching. If you have any questions, you may ask them in comment section. Please do give your feedback in form of likes and comments. Thank you once again for watching.